Hey everyone, welcome to Neura Pod. Our channel covers all things related to Neuralink, but keep in mind Neura Pod receives no compensation from Neuralink and is not formally affiliated with Neuralink in any way. Here's a clip of former Neuralink president Max Hodak from the first public Neuralink launch event. This clip will set the stage for the rest of the episode.
I just want to talk about briefly that Neuralink didn't come out of nowhere. There's a long academic heritage of research here. The cochlear implant has reached millions of patients since the 50s for deaf patients. Over 100,000 patients have received deep brain stimulation for Parkinson's and essential tremor and dystonia and now other indications. And about 20 patients have received the Utah array, which is a little 100 electrode rigid metal silicon device. And even though it has very few channels, they've been able to do some really cool stuff with it. There's videos on YouTube of BrainGate patients doing things like controlling tablet computers or even texting each other through Utah arrays just from the small number of electrodes. And so many of the people on the team at Neuralink came from this academic work. I got my start working in a lab at Duke University studying how mappings between brain and like the screen space change. So if you make it so that the joystick goes, like the cursor goes sideways when you push forward instead of up, like how does the brain change that representation? So the point is that there are lots of people that have been looking at this problem from lots of angles for decades, and we're in the greatest sense building on the shoulders of giants here.
The Utah array that Max references has a small number of electrodes compared to what Neuralink is aspiring for in the long run. However, at the time of development, the array was 2 orders of magnitude higher than had been possible for monkeys or humans. One of those people who played a key role in shaping how the field progressed from the 1990s to now is Dr. John Donoghue. BrainGate's website has this to say about him. Quote, "For more than 20 years, Dr. Donoghue has conducted research on brain-computer interfaces, and his laboratory is internationally recognized as a leader in this field." Unquote. For those who haven't seen our prior episodes, BrainGate is interacting with Neuralink. Listen to this clip from the head of BrainGate, Dr. Lee Hochberg.
You may have seen in the initial disclosure side that Mass General has a clinical research support agreement with 3 companies that are relevant in this space, if not more, which includes Neuralink, as you just mentioned, Paradromics and Synchron. So I'm very fortunate to have the opportunity to interact with them every so often through the hospital, because what we're learning in our BrainGate research, I want them to know.
There's a young guy playing a video game. It's called He-Man. There's treasure chests here and goblins, and gamers know that they have another name, but I always forget it. So he's supposed to hit these treasure chests. So he's doing an okay job. That if you know any 20-year-old kid that's a gamer, that's not particularly great performance. So this is Matthew Nagel, and Matthew had what's called a C3-4 a complete spinal transection. He was stabbed in the neck. He has no connection between his brain and his body because his spinal cord was transected. So he has this BrainGate implant that I'm going to talk about in his brain. It's a small chip that's got 100 electrodes in it. That the electrodes are bringing the signals related to his intention to move his arm out to the outside. That's being fed through a computer where algorithms are decoding it. And the decoded input is fed into a computer, and the computer thinks it's getting a mouse input, and so he's playing that game by thinking about moving.
There was a moment of true joy, true happiness. I mean, it was beyond the fact that it was an accomplishment, I think an important advance in the entire field of brain-computer interfaces, that it was really a moment where we helped somebody do something that they had wished to do for many years. This is the culmination of more than a decade of work, beginning with science at Brown University, that laid the foundation for all of the translational work that led from monkeys playing video games using their thoughts to humans being able to control a cursor in 2006 to ultimately to today's accomplishments.
So clearly BrainGate had and still has a significant influence in progressing the field of brain-computer interfaces. In 2015, when he was asked about what BrainGate is working on, here's what Dr. Donoghue had to say.
So this BrainGate project that we're working on is literally to reconnect the brain to the outside world for people who are paralyzed, people who have had that connection broken, either by injury or by disease, such as a stroke or even ALS, as Stephen Hawking has.
For the past month or so, I've started learning more about the history of this tech. And after learning more about BrainGate and how it was formed, I realized that Dr. Donoghue and team had many of the same goals as Neuralink. I had the privilege of interviewing Dr. Donoghue, and in this short clip, he discusses the initial vision that pulled him away from academia to start a company called Cyberkinetics.
When I started and said we're going to create something, the goal was to take a person who is paralyzed and have them doing everything that you and I do every day, and you wouldn't recognize the difference. That would be my ultimate goal. I'm probably not going to see that happen, but if somebody does it, I'll be very happy.
And here's the rest of our interview. He first describes how Cyberkinetics got started. Hope you guys enjoy.
My interest had always been in how the cortex works, and particularly the part of the cortex called the motor cortex, which is a part that takes all the information that's going on in your brain and helps get it out to your body to make complicated, rich movements. And we knew in the 1980s, it was pretty clear that understanding what was going on with respect to movement would require recording from neural networks. And I did various things using wires that were not unlike what Neuralink is using today, but they only had one site per wire. And I was putting those wires into the brain and recording in the mid-1990s. And then I realized that this was a difficult and not reliable method that I was using, especially since I was putting them in by hand, not with a fancy robot. And so it was actually in the and I don't know, it was 1996, 97, sometime in that time frame, I went to the neuroscience meetings and I said, I'm going to find the best technology that exists so we can start doing multi-unit and array recording, multi-electrode recording. And I met Dick Norman at his poster, and he had this wonderful device, and we talked, and I said, let's try to make this work in motor cortex. And his student, Ed Maynard, worked between our two labs, one in Salt Lake City and one at Brown. And shortly thereafter, we published a paper in 1999 showing that you could get signals out of motor cortex and you could make sense out of these signals with respect to movement. But others, of course, had already, Apostolos, Georgopoulos, and other people had already said that if you take populations of neurons, you should be able to, you can decode what's going on in motor cortex. But nobody had taken them sort of all at once. So we showed in 1999, we showed the array would work. And Misha Soroya in the lab, along with Nico Hatsopoulos and others, then said, well, let's see if we really can decode in real time, then we can be able to read out a monkey's brain and have a monkey control a cursor and play a video game. And that was Misha's PhD thesis. And that was in 2002 that was published. So it was actually surprisingly a short time later that we had direct demonstration of being able to use a brain to control devices, and that it was, safe and it would work for a period of time. We didn't know at that time how long it might or might not last, but it was working long enough that we could do these studies and show that we could record many cells. So we said, here's our opportunity to be able to really help people. We said we could take the signal, we have the tool to get the signals in the brain. We have an understanding of how to make sense out of the signals, decode them. And we also can control things that people who are paralyzed can't do, like control a computer. So with a bunch of fortuitous events, we formed Cyberkinetics, which was actually with more technology people that were and engineers at Salt Lake City, part of Dick Norman's group, and people that were together in Boston, and some venture capitalists helped us out. So we were really in 2000, this was 2002. And by 2004, we were all ready to submit to the to the FDA and get an IDE of permission so that we could implant a person. And then shortly thereafter, we implanted Matt Nagel with a device and showed that Matt could play video games, control a robot arm, a very simple robot arm. If you look online, my favorite is he couldn't move at all. Matt was paralyzed from the neck down. He's a spinal cord injured patient with the inability to move at all. And he just wanted to move something. So we weren't very sophisticated at that time. We were just beginning to figure out, all the things we could do. So we got a prosthetic hand that could only open and close the hand. We laid it on a blanket on his lap. And we connected up a decoded signal and said, okay, Matt, you can control this. And if you look online, you can see the movie. And so he opens and closes and he goes, holy shit, you know, those are my favorite moments that he was able to move it. But in addition, we had him draw circles and, you know, do spelling, use email, things like that. So he was really, you know, controlling devices right out of the box. And probably the biggest limitation then was the control of the cursor was, you know, sloppy, I would say, you know, a little bit slower, a little and less accurate. And so that was Cyberkinetics was busy doing those kinds of things from 2004 through 2008. And the problem was that although we had raised a bunch of VC money, that we really weren't ready with one of the things, you know, we had on the table a fully implantable device. And it was in 2008, there was a big crash and, you know, the money that was necessary wasn't there. And that was basically the end of Cyberkinetics. They just couldn't sustain themselves. So two big events happened then. Then BrainGate, the research study, came back into academia. And Lee Hochberg is now leading that consortium, which includes Krishna Shenoy and Jamie Henderson at Stanford and Bob Kirsch and Balu Lajaboy at Case Western. And those guys are, that's an effort today, you know, the research, the IDE trial that's ongoing now is the sort of clinical work that's been, you know, making breakthrough after breakthrough on what you could do with the brain-computer interface. And at the same time, the sort of technological components of Cyberkinetics was picked up and became Blackrock Microsystems. And Blackrock makes the devices, they make the arrays, they make the amplifiers and the devices that people are using. As far as I know, virtually all the human studies that are going on in the world that are using array recordings are using Blackrock's technology. So that was so Cyberkinetics, although it had a sad end, it did give rise to two very important things: the BrainGate consortium that exists today, doing research and development, and then the Blackrock, which is supplying the field with very important technology. So that's sort of in a nutshell what's happened over many years, and I've been involved. Both in those early stages, the formation of Cyberkinetics, and then continuing on, especially with decoding kinds of activities and understanding how to make sense out of signals in the brain.
Gotcha. Yeah, there's a lot of good stuff in there that I want to go into further detail with you about.
Sure.
So one of the things that I had found in the research was essentially people are crediting you and Cyberkinetics with the first implanted Utah array. Is that correct?
In a human.
Sure.
Yeah, in a human. Yeah.
And would you say that that's true? Like, do you feel like you guys were the first ones to do that?
Well, I never like to claim anything first, but I don't know of anybody else that's put the Utah array in a human. The Matt Nagel was the first human to have that device. Of course, Philip Kennedy had put a different kind of electrode, a few of them, into a couple of humans in the around 2000, a little bit before that, with just a few channels, and had an early version of a brain-computer interface, which was wireless. So that was an early start that he pioneered. And then before, you know, so you said in humans, and I'm virtually certain that nobody else had put an array in a human before Matt Nagel, his implant. But prior to that, of course, Dick Norman's lab, who developed this technology, had put it in animals and was developing it. One of the main things they were going to do with this technology, and there's still some interest, I think it's been a revived interest, is to use it to stimulate in the visual cortex to be able to recreate vision. So in this case, it's not recording and reading out the brain, but stimulation of the brain to recreate patterns, which I think it's a really good idea to be able to help restore vision to people who can't see.
I see. And so I guess in line with that thinking, if I or whomever is trying to explain to a regular member of society that doesn't really know too much about all this, then does it make sense to call these brain machine interfaces basically like pacemakers, but for for the brain rather than something that helps regulate like a heartbeat. It's helping regulate different signals in the brain.
No, because I think you want to make a very clear distinction. There's two different interactions with the brain that neural interfaces can do. They can read it out or write in. So let's talk about reading out. Reading out is listening to brain activity, picking up brain signals, and trying to understand what's going on in the brain. It's reading, like reading a book. You're looking at characters, and you interpret those characters as words and then give it meaning in sentences. Here, you're looking at neural activity and reading it out. So why are you doing that? Because people who are paralyzed create those messages in their brain. They create words for movement in a sense, but they can't get to your muscles because the pathway is broken, right? So either in the brain from a stroke, or maybe in the spinal cord from an injury, or maybe the nerves are damaged going to the arm. But in any event, you create the signals and but yet they can't get to the muscles, so you can't move, you're paralyzed. And so in reading out, all we're trying to do is jump that gap, right? We go directly from the brain to whatever you want to control. You and I sit down and we pick up our computer mouse and we use the mouse on the computer with our hand. Our brain is controlling our hand that's moving the mouse. We just went straight to the computer and skipped the hand, which we couldn't make work again. Mm-hmm. So that's the readout part. You see what I mean? And you can read out sort of generalized signals with an EEG, which is sort of the up and down roar of activity in the brain, or you can read out the details, which the individual neurons have. They have detailed, complex messages that they're producing. On the other hand, you can stimulate the brain. You can activate it. And you mentioned a cardiac pacemaker, and there's a pacer where the heart's job is to produce a rhythmic contraction. In some people, it can't do that. It's something has gone wrong. So you put electrodes into it and you drive electricity out and stimulate the heart muscle and make it pump normally. Now, that same concept was used beginning in the 1980s to say, okay, maybe in a disease like Parkinson's disease, where you're shaking and you're stiff and the motor circuits are not working properly, you can sort of kick that motor circuit back into working properly by stimulating it. And in fact, that was rather remarkable that you could even do that, right? That you could put a rather large macro electrode into a small target deep in the brain and start pulsing it at 100 times a second. And now the thing starts working again. And it's kind of like, imagine actually now everybody has electronic emergency brakes, but in the old fashioned cars, when you pulled a cable that put your brakes on. Now, so deep brain stimulation, you can think of the brake is on in the car and you're trying to go forward, but you can't because the brakes are on and you can't release the brake. So you take a pair of snippers and you cut the cable, right? And now, so it's equivalent in one sense of cutting the cable to the brakes and allowing you to move again. And it's much more subtle than that. And it is by a very fortuitous chance that the very complex circuits of the brain that are sending information all over the place, you can kick those into action by stimulating. So that's been done for Parkinson's disease. And some of it's been tried for depression. So you can get a depression circuit that seems to also not be working properly, and you can kick it back into gear to work again by stimulation. That one is investigational. It's not an approved use yet by the FDA. And then there are many other uses, like in epilepsy, where again you can stop the seizure from coming on by stimulating. So that's injecting something into the brain, and they're really very different. You see what I mean? So you have to so it's read what BrainGate does, or write, which is what DBS does for Parkinson's and a bunch of other disorders. Gotcha.
And so the read portion is not similar to the pacemaker, and that's
no. So in fact, just to be clear, the DBS was actually called a pacemaker for the brain because it's stimulating. Oh, okay. Well, that's because the pacer, the little what's called an IPG, an implantable pulse generator, is virtually the same device for a cardiac device and for a DBS, and they're made by the same companies. And so you're pacing the heart, and which is some it's a slower frequency, and you're turning that into a frequency of pacing the brain by stimulating. And it really doesn't pace the brain as much as turn off the circuit, but it has been called a pacer for the brain. So stimulation devices have been called brain pacers.
Gotcha. Okay, understood. And so let's see, you talked a little bit about like the different technologies and how they got split up from Cyberkinetics to eventually becoming Blackrock, Microsystems, and now BrainGate. And I saw in some of the articles that you guys were able to license or some other way use different technologies that were considered proprietary. And I'm curious how you were able to get access to some of those like decoding algorithms or other types of proprietary technology.
No, there was nothing proprietary. In fact, I think I can say that we invented all the things. So there were two classes of things. Decoders were public. They were scientists like Andy Schwartz and Apostolos Georgopoulos or other people, Richard Anderson or things. They were publishing the way they were decoding information. So once it's published, it is in the public domain. Anybody can use it. We actually, working with Liam Paninsky and other mathematicians at Brown, Michael Black and Ellie Bienenstock, we were able to develop our own methods, and some of those we patented. So we actually held or hold patents on the ways of decoding. But really, we developed a lot of those, and we used our own device. We didn't have to get licenses from anybody. Okay. The other thing is when you're doing academic research, of course, people allow you to use even proprietary things. Usually it's customary to allow researchers to go ahead and use those kinds of things because you're not a for-profit business. You're trying to advance knowledge.
Gotcha. Okay.
So I can say as far as the electrodes, well, the electrodes were part of the University of Utah. So in that sense, we did license it from the University of Utah, but Dick Norman was part of the company. So that wasn't especially, it wasn't a difficult thing. As I said, so there was the technology, there was the decoding we had, and then the rest of the system we developed. It was all, and pretty much a lot of what we developed is still used in BrainGate or by everybody else today, all that hardware.
Okay. Gosh, I got to get my facts right. I've been like reading actually.
No, I realize that a lot of this history is not known. And by the way, I want to be very clear. I don't want it to come across that we were the only ones doing this. There were a number of other people working in the field and other people making important breakthroughs and contributing. And everyone was exchanging this information. There was an annual meeting, which is still held, but it was a different form of meeting. Every year, the neuroprosthetics meeting by Bill Heath-Dirks was a person at the NIH who was very important at bringing everybody together. He, in fact, helped to develop and promote the development of the cochlear implant. And Bill would bring everybody together and people would get together and talk about what they were doing and share information and say, where is the field going and what can we do? And so there was a lot of communication in the field, and that helped generate activity.
I see. Well, so a lot of these technologies that you guys were working on, you said basically the end of the 90s till 2002 or so. Can you talk about towards the end of Cyberkinetics, like what that was like and what it felt like to run a public company, what it felt like to go raise money, have investor calls, and just communicate to the public, like, here's the potential for the future, here's what we're working on now, and here's what we're building off of.
Yeah, well, when I started, I was very naive, and I had help in particular from a very bright undergraduate who's now a Caltech faculty member, Mikhail Shapiro. And we literally, he was only 20 years old at the time, I think, and the two of us would go around giving pitches to people. And most people, I think, thought we were crazy. But eventually we managed to get venture capital. We got some investment, and then it was a company, and I was not the CEO. There was a real CEO and a whole staff of people, and we actually did eventually go public. to try to sustain the company. But as I said, I do think cyberkinetics was very early. I mean, I'm excited to see things like Neuralink, which is pushing the field forward, and beginning to realize what was my dream of successfully providing this technology to people who need it. It was very hectic, to run a company in those days. We both had to convince people of the merit. We had, I think, very compelling results. One of the things that was very interesting is we showed that people that had strokes, people that had spinal cord injury, and people that had ALS, which is amyotrophic lateral sclerosis, where their motor neurons degenerate. They're all completely different ways of becoming paralyzed. But in every case, those people could control computers with their brains. And so that was a really important medical discovery, I think, that to say that there was promise for everybody to be able to use brain-computer interfaces. So I was very sad when that we couldn't sustain this because it as now everybody knows it's very resource intensive, and simply research grants alone are not enough to sustain the activity. Clinical trials are very expensive, they're very complex, and the technology is expensive and complex, and you need to bring together clinicians and engineers and neuroscientists all working together to make the product actually suitable for patients, for people that need them. And that's a really big effort. And, those groups are not used to working together. And that was one of the things that we worked very hard on, having a culture that promoted, people from the business world and people from the science and academic science, engineering, and clinical world all working together for a common purpose. And everybody in cyberkinetics was really dedicated to the goal of really helping people.
I see. Well, so I don't know if you've been following recently, but there's a lot of talk in the news about SPACs and different companies that will merge with other companies who truly do not too much. And then it's a quicker way to access the public markets. And it sounds like that's essentially what you guys did.
Well, I'll tell you that actually, yeah. So that was probably a mistake, but okay. we actually did. It was then called a reverse merger. And we became, I think, a few hours, Trafalgar Mines, because we reverse merged into Trafalgar Mines, which made us public. But once you become public, there are so many regulatory requirements that you eat up huge amounts of resources to run the company. Accounting is much more strict. And so the company was basically, hemorrhaging money to manage all the financial regulatory requirements. And I think, startup companies should stay private as long as they can, even though the public markets can generate a lot more revenue. And also, I think, the fact that we were in the 2008 timeframe when there was a big crunch in the markets, there were no roots left to raise the money that was needed.
Gotcha. And up until going public, how did you guys raise funds?
It was VC money. We had investors that was all venture money investors who we raised quite a bit of money. I don't remember the total amount we raised, but it was quite a bit. And but it was still small even by today's standards. It was small in terms of what we needed to get done. Which I think we had created an array, and so we were able to record signals very nicely. And use those signals to control things, but we did not have a fully implantable device. And that's, without it being wireless, it's difficult because patients need to move around in the world. And having the BrainGate device we're using then, and still to some extent until the paper you saw recently about wireless stuff, with it being wired, you've got these big cables connecting to the connector on the head, and that's really cumbersome and not suitable for use in the home. Now that the new paper that Hochberg and the BrainGate group published show that you can have, even if you still have a pedestal on the head, that people can actually function in their home environment using this device. So that's a big step towards making sure that, or making progress towards a suitable device. All of these advances are important to having something that can be used by people to help them, live a more independent life and do what they want to do when they want to do it.
And I assume that this was something that they were working on prior to COVID, but now that COVID had kind of come around, it probably accelerated that effort to make sure that the wireless.
Yeah, nothing in this field happens in a year, right? It's a very long. So this really is part of. So and that is me working with Arto Nurmiko and with Lee and others, but really it was Arto Nurmiko and his student Dave Borton, who made the first high bandwidth, fully implantable device. I don't remember the year they put that up, but this now 7 or 8 or more years ago. So that's a 100-channel full bandwidth implantable device. And that device, so they built it, they tested it in animals, and it's sort of available to be finished for humans, but it's not been in a human yet. Okay.
Yeah, I mean, when the article came out, I was pretty excited because I think that, it's just like any progress that can be made towards this grander vision is just super cool.
So what I didn't finish saying, just to finish, is the device that was in the paper shows a device that is screwed onto the pedestal, the plug on the head. So that's a little wireless device. It's a tiny device. And the reason it's a tiny device is because it is the electronics for the fully implantable device. And so this was a step to show that it worked. You see what I mean? So the implantable device has electronics inside a can. And basically, this was the electronics without the can on the head, but not inside. So it was a step in the right direction, but it's the same electronics that will be in the can for high resolution recording.
Okay. So what is the like end result? What's the vision with?
A fully implanted. Well, the next step would be to implant that system under the skin, and that it would be a fully implanted system that's totally wireless. So with the current device, the paper that just came out shows that the electronics that have been produced will work. They will record signals, they will record high bandwidth, and they will transmit them wirelessly into the environment where they can be picked up and converted into control signals. And a human can use that system.
Which in theory is possible. You just like put this thing that's on your head and then you put it inside, and in theory it all works the same, but I assume that's
not that easy. No. Yeah. there's always something, you have to be very careful to make sure. And what has everybody done? They start with, on the bench, can you make the electronics, right? Step 1, can you build it? Step 2, can you put something in an animal model to see whether it does what you think it does? Does the electrode array record neurons? Can it pick up the signals? Does the circuitry work the way you think it does? Does it work in the environment to, when there's lots of other stray signals around, can you detect it? So the usual approach for something like a human device is to do this in a stepwise fashion, engineer it, test it in an animal model, make sure it's safe, which is first and foremost, and make sure that it does what you want it to do, because you don't want anything to go wrong and, to ever place a human in jeopardy in any way. So you want to be extremely careful that these devices are safe, first and foremost.
Sure. So in the latest update event, I think Neuralink announced 1,024 electrodes. And I'm curious what your take is on like how much more access to information and data they'll be able to get, and what do you think are some of the possibilities that they'll be able to unlock that some other companies have not been able to do?
Well, I don't think that's a company issue. That's a science question. I think the question you're asking is, how many channels do you need to understand the brain? Is that what you're asking?
Yes.
Yeah. So that depends on your theory of how the brain works. If it's 5 binary switches, you need crude electrodes and 5 of them. But if it's millions and millions of channels working together, you might need millions. But I think, you know, the people are not going to have millions of wires inserted into their head. Now, people may disagree with that, but I actually don't think that that's a practical solution. Mm-hmm. We actually are working on a solution that I can't talk about that I think is incredibly clever. My student is working on it, my collaborator now, and he's got a clever way to get around all this. But what
a tease.
Absent that, I think that, you know, what we do know now is that lots of neurons work together, and they do interesting things when they get together, just like, you know, football teams, soccer teams, crowds of people, you know, lots of interesting things happen, sometimes unexpected things. When groups of people or groups of neurons get together. And now the question is, how many of them do you need to sample in order to reveal what's actually going on? So the answer is certainly more than one. And, you know, the answer could be, if you really truly want to know everything that's going on in your brain right now, you might need to record from every neuron. But I don't know how to do that. I don't think anybody does. You know, there's no way to do that. So the really interesting one is, how many can you sample to get a sense of what's going on, right? Mm-hmm. And we know now that with you know, we now use 100 or 200, and some people have used up to 1,000 electrodes with the Utah Blackrock arrays. But typically in people, it's 100, 200, maybe 400, or up to 400, I think, electrodes. And those, you can really learn a lot about what's going on. You know, we showed in monkeys that you can decode from one little patch of the brain, you can decode the entire action of the arm. So you can, you know, the fingers, the hand, the wrist. It was a very nice paper in 2010 by Carlos Vargas Irwin in my group that showed that you can decode everything about the hand, not perfectly, and it's in real time, it would operate slowly, but it's all there. And so I guess you need more neurons to maybe make it faster or to make it a little more accurate. But I think that's a very important scientific question of how many neurons do you need? And we don't really know. But I know a few. What's surprising is, for any particular movement, like if I just want to open and close my hand, mm-hmm. if you catch the right neuron, you could do it with one neuron. That's all you'd need. But typically, you know, because the one neuron might not be so reliable, just a dozen or 2 dozen or 3 dozen would be enough.
I see.
If you want to do more things, you need more neurons.
Sure.
Yeah.
And do you think like a pathway to finding out what that answer is, to really having a better sense of how many neurons are required to have specific detailed output, do you think that can be found out by just trial and error over time? Like somebody puts in and is able to record from a certain set, and then you just see how the human behaves, or see like...
Oh, no. So let's, back up. So the question is, can you answer this question? First of all, you don't have to do this in humans. For example, Sergei Stavitsky and Eric Troutman are working with neuropixel electrodes. Each electrode there has about 1,000 sites per electrode. And I think at any one time you can record 394. And you can put those into the brain of a monkey, and you can record at a density higher than anybody's ever recorded before with these neuropixel electrodes. And with that very high density recording, I think you can get a sense of the nature of the code. How densely do you have to sample? That's what they're working on, you know? And so we'll know the answer, I think, in the next few years of what density is necessary to give you the kind of information you need to make a very successful brain-computer interface. So that can be done in animals. I mean, all of the animal work, the work, especially the monkey work, is extremely important to lay the groundwork so we know what's going on. So that when we first showed that humans could control cursors and type and move robots, all of that was based on principles that we had learned from able-bodied monkeys. Basically, you know, we call it playing video games, but doing things, you know, mm-hmm. doing complex motor behaviors. We learned all the rules. I think if we had strictly gone in the dark and started recording in humans, we wouldn't know what to do.
I see.
You know, we'd be lost. We had to have that important scientific foundation behind all of this before we went in. So when we went in, we knew basically what to do to make sense out of the signals.
Gotcha. So I'm trying to, okay, so I'll ask you a question about like where you're currently at right now. Are you formally affiliated with BrainGate still? Or do you just
yes,
talk with them?
I'm in a, so I spent a number of years recently on a leave from Brown, helping to set up something called the V Center for Neuroengineering in Switzerland. It's part of EPFL, which is another leading place in development of neural prosthetics. They're working on things like devices to restore locomotion, walking in people who are paralyzed, and Gregor Cortin's group and a bunch of his very talented collaborators there are all working to, with really remarkable success to help people walk again who have had spinal cord injury. And that center was set up to help promote that research. And I was, honored to be able to go there and help set it up. And it was fun to live in Europe and then fun to interact with a different group of scientists because I had been at Brown all my career. And then I had agreed to do that for 5 years and then come back to Brown, which I was very excited to do. So I did that. That's all set up and running. It's a wonderful place. If you ever get to go to Geneva, Switzerland, you can see this place called Campus Biotech, which is one of the most magnificent neuroscience centers on Earth. It's really beautiful. And it's also a hotbed of science. So I came back to Brown after I set up the center there. And so I'm, Lee Hochberg is really the person who is running and organizing and maintaining a very complex consortium that's really, he's doing an excellent job of running that collection of, as I said, Krishna Shenoy, Jamie Henderson from Stanford, Balu Ajaboy from Case Western, really superb people. And they work together. And the series of findings that have been emerging over the past, 5 years or so, improvements in decoding, better control, rapid communication, spelling, those kinds of things, typing, all those advances have come out of that group. And so I'm back as a member, which is kind of a fun position to be in. And then I have my own lab, which is using non-human primates as well as working with the human group I'm back to sort of understanding how the brain works. How does the cortex make you smart? How does it function? And then also how to apply that information in improving brain-computer interfaces. So I'm contributing in various places in that whole effort.
And that's pretty standard for what a member would do?
Well, yeah, some people are leaders in running human trials, so specific things like testing the ability to improve the rate of communication when somebody is typing, or communicating by spelling, or controlling their own arm. As you know, in 2017, I think we showed that a person can control their own arm by using the signals from the brain and connecting it to an FES system, a functional electrical stimulation system, basically stimulators in the arm, and move their own arm. And in fact, that when I started and said we're going to create something, the goal was to take a person who is paralyzed and have them doing everything that you and I do every day, and you wouldn't recognize the difference. That would be my ultimate goal. I'm probably not going to see that happen, but if somebody does it, I'll be very happy, because that was my goal. And it actually started with a discussion with Hunter Peckham, who really was the person who created the wonderful environment at Case Western. And it is still working on FES systems, that is systems that stimulate nerves and muscles and help people move their limbs. And at that point, he didn't care where the control signal came from, if it helped the person walk or it helped the person move their arm. They even had a woman who could wiggle her ears and trigger movements, you see, of her arm, by taking the muscle signals and using those as commands. And so we said in the 1990s, we're the perfect match. I want to control the body with the brain, where I'm the brain guy, and you want to control the body with stimulation, and you're the muscle and arm guy. So let's put it together and make it all work. You know, I'll do the brain half, and you'll do the body half. And so it wound up in 2017. We had the proof of concept of that idea that we sort of had in an informal conversation in the 1990s, which is kind of scary to see how long it took to happen. But it's a really big deal to watch that video of a person thinking about, you know, reaching out and grabbing a cup and taking a drink on their own when they can't, and they did it. And so I think that's now on the path of happening. If it'll, you know, I want to see it so that that person can do anything they want at any time. They can go play basketball, or, you know, go for a walk with their family, or so, but that's, you know, still quite a ways off. And there's still a bunch of technological feats and scientific advances that are still need to be done to get to that point.
Gotcha. Yeah, I think I saw somewhere one of the initial goals was to have somebody be able to play basketball after they were paralyzed.
Yep. Yeah, I used to show up in beginning of my talk. I used to show Obama playing basketball, and I'd say, I want him to play basketball with somebody who's paralyzed and not know they're paralyzed. Yeah,
it would be incredible. It's like, well, I think that's something that I admire so much about what Neuralink has, what their vision is that they want to make this truly scalable. And Elon just has this, I don't know, crazy mindset that he can get anything done. And then it's just like, well, if they can do it with one person and use a robot to do it, then in theory, that just scales up so significantly.
Well, I think that's what everybody in the field has wanted. Is again, so it's sort of, I think you could say, and I think you can argue this, but somewhere in the early 2000s, you know, a whole bunch of people said, you know, we really need to, you know, we really need to apply what we've learned to make it possible for paralyzed people to move again. And that's what we wanted to do, is create a device that there could be hundreds of thousands of people benefiting, not the whatever it is now, 20 or so people, you know. So that was the scaling idea has been around for decades. But it is really important that people who are visionaries, people who have the ability to push the field forward, enter and point out to the public how important it is to invest, but not only at the commercial end, because they'll make an important commercial product, I hope, one day, but there are many, many other applications, and we need a way to be able to ensure that all the potential of this field, everything that's possible, is done. And right now, my biggest worry is we don't have a way to accelerate the development of devices. We need more, a way to sort of ensure that, many of the ideas that people have now about using technology to cure mental health disorders, neurological disorders, that those actually come to fruition. And I think it's going too slowly now. We need, you know, 10 Neuralinks, you know, with all working on various applications. Mm-hmm. Neuralink has their idea. We need other people working on other applications. And there are, but most of them are in research labs. And those are important, great people, but we need something to help push it faster. You know, we need something that will drive it even faster and further, so that more people can benefit by this. You know, we've had pharmacology for whatever, 100 years, a couple of 100 years of using drugs to treat nervous system diseases. The progress as of late, arguably, has not been as fast as we'd like it to be. But now we have a whole new domain of ways of helping people using technology. You know, actually, one of the things we used to joke about in the lab is we'd say, we're in the replacement parts business. You know, the brain is broken and we're going to build you a replacement part. Your car breaks, you know, your generator doesn't work, and so you go get a new generator. Well, you know, your motor system doesn't work. We're going to make you a new motor system that's going to be made with physical components, not with biological ones. Mm-hmm. And I think we are getting to know how to do that. That we've shown that even the kind of technology we have today is good enough to really help people.
I thought it'd be cool to include this as well. After we finished up, I told Dr. Donoghue we'd show him the edited video prior to release, and he shared these thoughts.
My biggest worry is that, well, not my biggest worry, but a worry I have is that somehow when you clip things the context changes and people misinterpret it, and it's a little bit of a sensitive time, you know, that people are sensitive, you know, like I'm especially, you know, that some people think, oh, you're claiming you did this before anybody else. And, you know, I think so-and-so did it first. And I want to be clear that, you know, everything that's happened is always a group effort. You stand on the shoulders of each other in a sense, and that we don't claim that we made everything happen. We're just part of the, you know, the whole large enterprise of scientists and entrepreneurs and trying to move things forward. And I think it's just a matter of sometimes you inadvertently clip things and you don't understand the sensitivities in the field. And we want to make sure we want to create this inclusive environment where people feel like they all should be communicating, they should work together, there should be a spirit of exchanging information. And I think that's what we're trying to create. I just want to make sure that there isn't any misconception that can emerge, because I've been in the press a lot, and sometimes people come back and say, why did you say that? You know, I say, well, I didn't think I meant that when I said it, but, you know, they misunderstand. So, and it's a very important time to do that, because now there's a lot of attention, you know, a lot of activity. So we want to make sure that we give a very positive message about how good all this activity is and how important everybody's efforts are to really doing something important for people.
And just a reminder, Neura Pod receives no compensation from Neuralink and is not formally affiliated with Neuralink in any way. Thanks again for tuning in. Hope you join us again for the next episode.